Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and a very good morning to all. On behalf of Apta Niaga Sdn Bhd, I would like to our heartfelt appreciation to Dr. Aswir Abdul Rashid, Presiden Kelab IMR, Ketua Unit Pemakanan NMCRC IMR, dan semua organizer dari Kelab IMR dan Unit Pemakanan NMCRC IMR. I'm Shafika, the co-founder of Apta Niaga Sdn Bhd. I would like to, it is with great pleasure that I welcome everyone present here, joining our meaningful sharing on this particular topic, exploring next generation sequencing, microbiome diversity to insights. Sequencing refers to the huge amount of DNA molecules that are able to be read at the same time. So this actually encounter the first problem of the first generation of sequencing. However, in NGS sequencing, besides the same library preparation of the denaturation of DNA, actually the NGS sequencing also can be used for RNA. So for RNA, it will be reverse transcribed first into cDNA, and then the cDNA will be, it can be used for the amplification using PCR. So there comes the limitation of the NGS sequencing, as it will cause amplification bias.
My name is Muhammad Nazri again, Muhammad Nazri Abdul Bahri from Apta Niaga Sdn Bhd. Okay, you have learned from Dr. Fika just now about the principle, the application, the history of NGS, and my part is to cover the recent advancements of NGS in microbiome studies. And then we recommend you to have a mock microbial community standard. This microbial community standard is a synthetic microbial mixtures with precisely defined composition of non-microbial species or strain. It is designed to mimic the natural microbial communities. So you will see single cells and single bits in one capsule. So from here, the next step is the lysis buffer inside the microparticles will lyse the cells and then expose the RNA into the microcapsules, and this RNA will hybridize with the adapters inside the capsules.
Yeah, I'm just wondering, like when they prepare the biofertilizers, is it consistent, like the microbiota will be consistent between this preparation and then the second or third preparation or something like that? Do you guys have any answer to that?
So actually, I think that is for the quality control of the products, in case they are producing or they are manufacturing their own biofertilizers, which can also be carried out by us. In this case, our customers, they send us their soil sample because they are doing like pre-planting measures. So they want to know if their area of plantations are contaminated with pathogenic microbes. So based on the Corona and based on our own database, we found a few pathogenic microbes and also beneficial microbes. So these pathogenic microbes are actually can cause diseases specific to the crops itself. So for example, Ganoderma boninense, they can't cause disease to the cucumber, for example. So Ganoderma boninense are only cause diseases to the oil palm. So for our customer, because it is actually confidential, we cannot tell you, but the pathogens that we discovered are not giving the risk to the crops that they are going to plant.
The microbe, is it doing the are you doing the genetic modifying the microbe, and then is it just now that you say about the transplant? Is it transplant to the patients?
Okay, those microbiome engineering that I share with you are actually based on articles and published articles. We as the molecular service provider, we don't actually provide that kind of services. However, we did molecular cloning that can help on the transformation of these genes of interest that you like on host bacteria, something like that. But coming back to your questions, actually, our lab personally didn't do that kind of.
But that is happening overseas.
It's from the literature, but the clinical they are still in the stage of clinical trials, and I think the only microbiome engineering that have been approved by the FDA is on the fecal microbial transplantation. Only that.
So what I mean is, I think that's become a trend in the future. Maybe the antibody will be replaced by some GMO microbes.
Those are some strategies that you can approach for your projects.
Thank you so much.
Thank you.